everyone, I'm Laura Dormer and I'm the editor of The Evidence Base. I'm joined by Dr. Lewis Carpenter from Arcturis, who's going to be sharing his expertise on external control arms derived from real-world data using anonymised NHS electronic health records. So welcome, Lewis. And to kick things off, please could you introduce yourself and tell us a bit about your role at Arcturis?
Hi, Laura. Thank you for speaking with me today. So yes, I head the real-world evidence function here at Arcturis. I'm a medical statistician by training, and I've had a long history of working in both academia and industry. So Arcturis is a company that partners with NHS trusts under strategic research agreements to access anonymized electronic health records. We foster collaboration with the clinical and data teams at each trust to collect high-quality real-world data. And my job at Arcturis is to head the real-world evidence team to analyze this highly curated real-world data and provide real-world insights to life science industry to support clinical development programs right across the clinical development pathway. This includes from drug discovery and target product profiles right through to long-term safety and efficacy monitoring post-authorization.
To give us a bit of background, what is an external control arm and when are they used?
Yeah, that's a really good question, Laura. So I think it's first worth outlining what a randomized control trial is before going into external control arms. So randomized control trials will randomly allocate patients to either the interventional arm or the control arm, and that control arm can be a placebo or standard of care for which the intervention will be compared to. Now, randomization is an important component when determining whether a treatment is effective or not, since a well-designed and conducted randomized control trial will ensure balance of both measured and unmeasured confounders across the two arms. Therefore, any difference observed between the two arms can be attributed solely to that new intervention. However, not all clinical studies do include a randomized control group. For example, early phase 2 studies are typically single arm only, so they'll only provide the intervention to a patient group. And there are also some instances where recruitment of a control group in a randomized control trial is either ethically or practically not possible. Now, there's been increasing interest from regulators such as NICE, the FDA, and EMA on how to leverage real-world data to derive the treatment effect of new therapeutics in those instances where a control arm is not possible. This has been a key focus of Arcturis and our real-world evidence offering, and this is where external control arms come in. So an external control arm refers to the development of a comparator group or control group for a clinical trial where the control arm is not randomly allocated within the trial itself. Instead, the control arm is generated from external data sources, and this could be control data from other completed historical clinical trials, retrospective cohort data, or in this case, electronic health records from healthcare systems. For single-arm phase 2 studies, this external control data can provide pivotal evidence on the efficacy of new treatments earlier in the clinical pipeline, as well as providing vital evidence on the treatment efficacy where a clinical trial is not possible due to those ethical or practical considerations. And these can be used as part of an evidence review component for a health technology appraisal to regulators such as NICE.
What are some of the key components to a successful external control arm?
That's a great question. So the success for an external control arm study is similar to all other scientific studies, really. And that requires adequate trial planning, transparency and open communication, and high quality data with appropriate statistical methods. Now, it's really important to outline that fully randomized trials are always preferred when it comes to providing treatment effects of new therapeutic agents. As such, it's critical that the use of an external control arm in lieu of a randomized control trial is fully justified. And this is where open communication with the regulatory bodies is really encouraged to identify that the use of an external control arm is fit for purpose. Transparency through the use of publications of study protocols to public registries helps ensure and foster trust and scientific reproducibility with your external control arm work. And here at Arcturis, we're strong adopters of clinical trial emulation approaches to when it comes to planning external control arm studies. And this is important to determine what your patient population will be, what the key clinical endpoints of interest are, what the observation period and index date are going to be, and any key confounders that you want to include in your statistical analysis to ensure that you can balance between the two groups. This planning will guide the exploration of suitable real-world data and ensure that data are fit for purpose.
What is it that makes real-world data regulatory grade?
So there's not real guidance from regulatory bodies such as FDA, EMA, and NICE on what constitutes as high quality real world data, but they do provide guidance around the type of aspects that they expect to see when it comes to high quality real world data. And this is largely around sort of three major components: a strong understanding of data provenance, a strong understanding of your data quality, and also that the data are relevant. Through Arcturis' unique partnership with our trusts, we gain a true understanding of the data provenance, and we can ensure that data quality through our feedback loop systems with the clinical care teams and data warehouse teams. And it's that allows us to close the gap and create what we call disease registries from electronic health records and allow the comparison between real world data and clinical trial data. We found through our work that there are usually quite a lot of trust level differences in terms of treatment pathways and coding practices that are used. And if these aren't correctly understood, it can lead to misinterpretations of the data. By accessing data directly from the NHS centres, this is particularly important for regulatory submissions to NICE, where UK data ensures that the data are generalisable to the NHS population for which the intervention will be introduced to. Given NICE's global reputation, high quality NHS data has the potential for much more global and broader use with other regulatory bodies such as FDA and EMA.
What statistical methods are needed to ensure robust clinical insights?
So, of course, in the absence of randomization with an external control arm study, it's important that the study analysis aims to reduce the impact of measured confounders between the external group derived from the real world data and the clinical trial data with the intervention group. Advances in causal inference frameworks allows us to combine those two different types of data, observational data and clinical trial data. During the study planning process, key confounders through that target trial emulation approach I mentioned earlier will identify which ones are needed, and this should be done a priori, typically in collaboration with relevant stakeholders such as clinical expertise, as well as what is published in the broader literature. Then, through the use of different methods such as inverse probability treatment weights derived from propensity scores and other methods such as G computation frameworks, you can begin to balance the differences between the two groups for those measured confounders that you've identified as being important.
And what are some of the key challenges when conducting an external control arm with real-world data, and how are the real-world evidence team at Arcturis seeking to overcome these challenges?
Yeah, so working with real-world data always comes with its challenges. So what we found in our sort of work that we've done so far is that whilst statistical methods are really good at handling measured confounders that we've identified through those a priori streams such as clinical experts and the literature, they are unable to deal with potential unmeasured confounding, and that's really where the randomization aspect of a fully randomized controlled trial is really quite key. So the use of new methods such as quantitative bias analysis are really effective tools at trying to be able to quantify what the impact of that unmeasured confounding might be on any derived treatment effect that you estimate. Missing data is also a well-known issue within real-world data sources. And whilst the first steps is trying to identify and reduce the missingness, and through our collaboration with NHS Trust, we look to try and understand which data are missing and whether those data can be completed. Other frameworks, such as multiple imputation, are really effective ways at handling missing data, and layering on different analytic frameworks through inverse probability treatment weights, along with bootstrapping methods for standard errors, on top of multiple imputation frameworks for missing data, really provide much more robust estimates for your treatment effect when combining real-world data with clinical trial data. Other biases that can remain is differences in how clinical endpoints are captured in real-world data versus clinical trial data. This was actually explored as part of a poster presentation Arcturis is presenting at ISPOR this year, looking at multiple myeloma patients. We're also looking at ways in which we can enhance the real-world data assets that we have from our trusts to really try to bridge the gap between clinical endpoints that are collected in trials through trial protocols and those that are collected in real-world data. This is particularly important in solid tumor indications where things like RECIST are typically used in clinical endpoints but are not captured in routine care. So can we look to do that retrospectively to bridge the gap between the two data items? We're also looking at novel statistical methods such as Bayesian statistics to combine clinical trial and real-world data more effectively. And this is depicted in another ISPOR poster we have this year, looking at Bayesian borrowing as a way of trying to accommodate real-world data with clinical trial data. Finally, real-world data is extremely effective at looking at particularly underrepresented groups that are not recruited into clinical trials. So again, we have other work at ISPOR looking at how underrepresented groups in advanced ovarian cancer could influence and have different outcomes relative to the clinical trial that we see in the published literature.
Great. Thank you, Lewis. And thank you for sharing your expertise on external control arms with us today.
Pleasure. Thank you so much, Laura.